Hello, everyone. I'd like to welcome you to today's webinar, OneShot, Advancing Forensic Biology with Integrated Genetic and Epigenetic Sequencing. My name is Paul Frere. I'm the NGS Field Application Scientist Manager at Twist Bioscience. I would like to give you a few housekeeping guidelines today. All lines will be muted during the webinar. If you have any questions, please submit in the Q&A box rather than the chat box, and we will answer all questions after the presentation. If you share your name, we will be able to get back to you after the webinar if your question has not been answered. Following the presentation, if you have 1 minute to take a brief survey, we would appreciate your feedback, and we will be recording this webinar. Now I would like to introduce you to our speaker for today. Dr. Mirna Ghemrawi is the Associate Director at the Center for Forensic Science Research and Education, or CFSRE, and Program Director for the MS in Forensics Biology at Thomas Jefferson University. She holds a PhD in biochemistry and specializes in forensics. Her research focuses on advancing forensic methodologies, including innovative techniques in DNA sequencing and biomarker identification. Dr. Ghemrawi is a dedicated educator, passionate about mentoring students and bridging research with practical forensic applications. She has received numerous accolades, including the Fulbright Scholarship and the Real Triumphs Award. It has been a pleasure working with Dr. Ghemrawi and her team on this project, and I'm happy to pass the mic to her to hear about her innovative technology. Dr. Ghemrawi, the stage is yours.
All right. Hello, everybody. Good morning for whoever is in the early time zone, and good afternoon for anyone who is on the other side of the world. It's such a pleasure today that I am speaking to you about one of the closest projects to my heart, the OneShot project. Keep in mind the name. We're going to get back to that name at the end of this presentation, but we're going to walk through we're going to talk a bit about the genetics part of forensics and the epigenetics part of it, and then how this project came to life. All right, so just in case you don't have a lot of background in forensic biology, it's important to touch base on what's the current practice in terms of active casework DNA analysis, or what we call conventional DNA typing in forensics. So the gold standard, if I would call it that way, it's basically looking at the set of markers known as short tandem repeats, and those tend to be different in terms of number of repeats between individuals that can be used for individualization purposes and identification. Obviously, if you're familiar with all of the shows you watch online, whether that's true crimes or some of the fictional with a caveat in how accurate these are, the application of forensic DNA typing is extremely valuable in different scenarios, not only active case crime scene investigation, but also paternity testing, or in cases of mass disaster, you have human remain identification. So the applications go on in so many different realms within forensics. However, the way it works, so when we generate these DNA profiles using these markers that I just mentioned, the STRs, we compare them to a database. One of the biggest databases known is what we call CODIS, and that has millions and millions of profiles. So that's compared oftentimes to that database in order to find hits in that database. But what if the suspect in this case, or let's say the perpetrator or the person we're trying to identify, has no profile, is not yet submitted to the database? Then we're not going to find any hit. Oftentimes, if this is the case, it becomes a cold case until that profile somehow comes to the database. So is there something else we at least can do as sort of investigative lead that would help in these investigations? And that's where some great researchers, some of them in Asia, some of them in Europe and in the United States, have looked at epigenetics or epigenomics. So a great paper, one of my favorite papers that I always like to read by Vidaki et al. Had made this graph just to show where the potential of applying epigenetics into forensics. So again, as a heads up, the epigenomics and forensics would ultimately bridge that gap between what's the fixed genetic data and the dynamic environmental influence. So it goes beyond just the genetics. And then particularly we focus, obviously, if you're familiar with the epigenetics research, there are different sub areas within that. What we're focused on, or the most studies that you can find in the literature, are particularly related to DNA methylation, or what we call CpG methylation. So these are like the cytosines that are followed by guanines, and some of these cytosines would have a methyl group on their 5' end. The research basically had looked at different applications or finding markers for different questions. Such as predicting age. So great researchers have also, other groups have come up with a set of CpG markers that can be used to predict the age of an individual. So if you think about it from a forensic standpoint, it is an investigative lead. If you're thinking of, a genealogy application, it's important to know, the age of the individual. It would be valuable in investigations. Another application would be also the body fluid identification. So again, if you're thinking about it from a forensic standpoint, getting a profile, a DNA profile in of itself is important, but that profile coming, let's say, from a saliva sample would have a completely different context as compared to DNA profile coming from a semen sample, which may mean that potentially a sexual assault had happened. So again, there are some CpG markers that are differentially methylated between the different tissue types. I like to call it more cell types that can be exploited for the purposes of body fluid identification. As mentioned about the age is another element that a lot of research has been done in that area, and some other phenotypic features like smoking status. Believe it or not, there are markers that can tell if someone is current smoker versus never smoked, or used to smoke and then stopped. Your methylation pattern can give us that sort of information. There are other studies that I'm familiar with that have done some studies on monozygotic twins as an example, but there are many, many other avenues and places where the research can expand to include other phenotypic features about alcohol use and abuse, illicit drug use, and other lifestyle type of information about an individual. So before jumping into the current versus how this idea came in mind, currently, at least to my knowledge, no active casework does any sort of epigenetics. It's still more so in the research side of the field, and that's because of multiple reasons. Oftentimes you're dealing with very minute, degraded type of sample when it comes to forensic exhibits. So the lab would want to save up as much as possible from that sample, only use it to the most needed type of testing. And the current methodologies in epigenetics would require some sort of conversion that would also require a lot of samples. So again, what's the current practice is, in case we're talking about human identification, as mentioned, we're looking at STR profiles. So if we're doing sequencing based, you extract your DNA, you prepare your library, amplifying those different markers, 20 to 27 markers, and then doing some sort of data analysis to come up with the DNA profile. If you're doing more on the age prediction, you have to have another extract of your DNA converted, and the most studies have done bisulfite conversion or a chemical conversion, and then the library prep sequencing, doing data analysis to get the CP methylation patterns and percentages to get the age prediction. And then you can easily, if you still have some of this bisulfite converted DNA, you can use that aliquot to also do more, because there's no one panel that can do both body fluid and age at this time. About both these types of CpG markers. So just by looking at the slide, that means we're consuming a lot of the sample, it's time consuming, it might not be too practical from a forensic standpoint. So is there a way where we can basically combine all of these together? So prior to last year, I would say probably it would have not been possible, because there wasn't a way where we can simultaneously look at both the genetic and the epigenetic markers in one sequencing run. But with the advancement of technology, and I'm going to talk more about the combination that we did, the two chemistries that we combine, we can do that. We can basically look at both the genetic and epigenetic markers from the same sample at the same time in the same sequencing run. And that's what we call the one-shot solution, followed by sequencing, and that's amenable to any type of sequencing platform, depending on what, let's say, indices you're using. And then there's a bioinformatics step here that's needed, that Biomoto call as base resolution, that I'm going to talk about in a minute, followed by some more data analysis to look both about the STR profile, so for human identification, but also get the CpG and methylation patterns for age prediction and body fluid identification. So how does it work? So I'm going to talk here first about what is known as Plus-Mod-C library prep by Biomoto, a company based in the UK, who came up with this great technology that would allow you to, again, being able to look at both the genetic and epigenetic markers in the same sample. So you start with your double-stranded DNA in this case, and then the first step that you do in this library prep is what's known as hairpin addition. So as you see in this graph, this hairpin is added to your double-stranded DNA, and this is just an illustration just to show that, for example, this C is non-methylated and this C is 5-methylcytosine, so methylated cytosine. The next step in this library would be to splice these two strands. So each of these hairpins will go with one of these strands, the sense and antisense, and then you will have these in your product. Following that step, there will be a copy strand synthesis. So this is just again, a polymerase that will amplify and make a copy. So now you have the original strand that has the epigenetics information. It's connected through that hairpin to that copy strand. Now the copy strand would lose basically the epigenetic information. Okay, and that's important because later on when we talk about the bioinformatic aspect of it, this is how, by comparing that, what we call R1 and R2, those two strands, we would be able not to only get the ATCG sequence, but also whether the C was methylated or not methylated. Following that, you have to obviously add the adapters and indices for it to be able to, so you can do the sequencing. So it's known as the fork head adapter ligation, and that is added to the strands. The next step would be to protect your methylated cytosines. So your methylated cytosines, it is protected, and through an enzymatic conversion, and keep in mind the enzymatic, because that's an advantage over the chemical conversion, your unmethylated cytosines will be converted to uracils. So now we have the protected, again, cytosines and the unmethylated cytosines converted to uracil. And then you amplify, basically, you make more copies of your DNA, and that's the library amplification step. So this is what the basically in a nutshell, in a very summary of the summary way, how the Plus-Mod-C library prep works. For probably a clinical or oncology or medical uses, this can go maybe after that through whole genome sequencing. You get a lot of data, and then you can do the analysis you need. Well, for forensic applications, that's not going to be possible for two main reasons, I would say. One is that oftentimes, again, you're dealing with very small amount of DNA, so you wouldn't be able to do whole genome sequencing. But the other part is about privacy concerns. So again, from a forensic point, the markers that we look at should not be informative of any clinical information, because that may breach the privacy of an individual. So we need a way, basically, to just look at what's forensically relevant. And that's where we needed hybridization capture to only look at these relevant markers. And that's where we partnered with Twist to come up with a panel that would fish or look at STR markers as well as CpG markers that have already been published in the literature that would tell us about age and body fluid. So the design basically was, we had two sets of designs. For the STR repeats, Twist had designed 528 probes to look at 26 STR markers, 20 of which are autosomal, and I think, if I'm not mistaken, 6 of which are Y STR, so only present on the Y chromosome, which again is valuable from a forensic standpoint. As for the other panel, which later on we just combined together, is the around 56 probes for 35 different CpGs, 15 of which are for body fluid, and 20 of which are for age prediction. These are the fluids that we focused on, so peripheral blood, semen, saliva, vaginal fluid, and menstrual blood. And the ones with asterisks also have CpG markers that are used for age prediction from these 3 different fluids. So, so how that hybridization capture methodology works is pretty straightforward and very easy to be done. So you have your libraries that I've just talked about, that plus mod-C libraries of your sample. You do the math to see how much volume you need to add based on the quant of this library together. So you pull these all, and then and remember, these are indexed primers. Indexed libraries that are enzymatically converted. The next step would be to hybridize your targets in solution. So we have these probes that were designed. They are hybridized with that custom panel that we've designed is added to our libraries. The next step will be to have this binding of the hybridization methylation targets to beads, so we can capture only the forensically relevant loci. And the last step prior to sequencing will be to do a post-capture PCR amplification and to purify and perform QC just to make sure that you have quality libraries that can move forward with sequencing. And for this particular study, since it's a small panel, we did the 2 hours hybridization. So again, quick way to do it. And for the post-capture PCR, we decided to start with 17 cycles to amplify our products. So this slide now doesn't seem too crowded, but in a minute it will be crowded. So I hope you can follow with me step by step about the proof of concept study that we did in collaboration with Twist and Biomoto. So we started with 2 individuals, the male who was 27 years old and the female who was 47 years old. The male, we asked them, and that's all IRB ethically approved. They signed a consent form, collected peripheral blood through a finger prick, saliva through a buccal swab, and semen sample that the male donor had provided. As for the female, she also provided peripheral blood as well as saliva, but also vaginal swab. And then we extracted the DNA. Since this is a proof of concept, we didn't want to go too low in terms of quant, so we've tested 2 concentrations, the 10 nanograms and 20 nanograms for this particular study. Since these are what we call reference samples, so like quality samples, we needed to fragment or sonicate our DNA. So we've used the Kuvaris M220 to sonicate our DNA to around 200 base pairs in length. So our fragments were around this size. Later on, once this is more optimized, we probably would be able to skip that step because oftentimes our forensic sample is around, if not less than that, fragment size. Following that, we did the OneShot solution, which is the +mod-C library, followed by capture. And one note in here, so for every sample we ran, we had spiked in controls, which are the PAC19 and Lambda. And those were very important for us to add because we want to make sure that the conversion that we did, the enzymatic conversion we did, was efficient in terms of sensitivity and specificity. So one of these is fully methylated, the other one is unmethylated. So we would be able to use them as our internal control to make sure that we have an efficient conversion. Then we did sequencing using the MiSeq. And the reason why we've decided to use the MiSeq for multiple reasons, one of which, this is what we had in the lab, but the main reason is that for it to be forensically applicable, we needed to use an instrument that you would find in some of the crime labs. And it's not higher throughput than a MiSeq, so we ended up just deciding to go with a MiSeq, and we used a microflow cell, loaded the libraries at 10 picomolar with 10% Phi-X. Remember I talked about the base resolution step. So for this one is, since the two fragments are connected through that hairpin, we would be able to compare the R1 and R2. Through this proprietary bioinformatic pipeline that Biomoto had developed, from the raw FASTQ file that's generated from the MiSeq, we get what's called as resolved FASTQs. And those resolved FASTQs will basically tell us all the 4 bases, so A, C, G, and T, and also will inform us about this modified cytosine, in this case, methylated cytosine. And since you are reading R1, R2, it has an inherent nature of it that it will identify and suppress any potential sequencing error, which also is an advantage in terms of accuracy of sequencing. Following that, once we generated the result FASTQs, we obviously had to do different types of analyses to, one, just look at the STR profile by looking at the flanking region around the repeat and counting these to make sure that they are the correct number of repeats as compared to the control. And then for the methylation, got the methylation percentages and did some math that I'm going to show in the next few slides. So I hope it was not too overwhelming and you were able to follow with me through these different steps. But now I'm going to talk about some general metrics, and then I'm going to show you some very cool results that we're really excited and proud of that we were able to get from this study. So some of this is not too relevant to everyone here in this room, but we had good cluster density around 14K, 1,400 K per millimeter square, and then good passing filter around more than 83% with relatively good Q30 scores. And the cool thing, remember that spiked in controls that I talked about? We use them to calculate the efficiency of conversion. We've had really great efficient conversion. So sensitivity-wise, we had more than 95, and specificity-wise, we had more than 99.8% based on the controls we spiked in. Now, the average coverage for the CpGs was around 30x. Some of them are more or less. As for the STRs, for most of them, we had around 10x. And remember, that was right from the first run we did. We had not yet done any sort of optimization. So as a starting proof of concept kind of study, we were very, very happy with the initial results that we got. So now looking at more of the awesome results. Okay, it's good to have coverage, it's good to have reads, but did it really work? So now I'm going to show you a result from an IGV screenshot just to look at specific locus. In this case, it's on the chromosome number 2. And I zoomed in to the particular coordinates where I know this marker on the ASIC4 gene that is a semen marker based on the literature. What the colors you see here, if it's blue, meaning it's hypomethylated, if it's orange, yellowish color, that's hypermethylated. So, and these are all of the samples that we aligned to the reference genome. And you can see that all of the female samples, blood, buccal, and vaginal, were hypomethylated. They're all blue. You have the blood from the male and the buccal from the male, like the female, were also hypomethylated. And the only sample that was hypermethylated is the semen, which means, again, it's proof that just by looking at this semen marker, and if we look at it, it's hypermethylated, then we know that we are dealing with a semen sample, and that would give an answer for the body fluid of interest that we're looking at from a forensic standpoint. Here I compile all of the data for all of the markers. So we had 3 markers per body fluid. And for all of the markers, they were hypermethylated for their corresponding fluid. So let's say blood marker that was hypermethylated for blood, but hypomethylated for all of the other fluids, with the exception of the 3rd marker in blood, which was the opposite. It was hypomethylated for blood, but hypermethylated for all of the other fluids. So just so we can walk through these results together, what you see here, all 3 markers for blood, and the samples, as you see here in the legend, the first 2 markers are hypermethylated for blood. So this is blood from the male, blood from the female, and then hypomethylated for all of the other fluids. Some of them, it's 0, so you don't even see them on the graph. So that means that these 2 markers did work. They are differentially methylated, and we can use them for blood identification. The 3rd marker, again, as I mentioned here, it's the opposite. So you see every other sample, so the salivas and the semen and the vaginal are hypermethylated, 50 and above, whereas the blood is hypomethylated. For saliva samples, you again have 3 markers, but in this case, all 3 are hypermethylated for saliva, but hypomethylated for all of the other fluids. It's like 70% and more, and less than 10%, if not 0, for most of the other body fluids. For semen marker, again, we only had 1 semen sample, and all 3 markers, it's like really hyper, like 90 and above, whereas all of the other samples was in the hypomethylated realm. Last but not least, vaginal fluid is in that methylation, these for all 3 markers, but all of the others are hypomethylated. And you may question, oh, why this is too low? It agrees with what the literature had shown. Vaginal sample is really a complex fluid. It has a lot of different cell types. That makes it more like more of a mixture of cells. So you wouldn't, especially for the markers that so far has been identified and validated, you're going to see it in that mid realm here, around 30 to 40% methylation. So all body fluids were correctly identified in this study. So we were very happy with these results. Now moving on to the age estimation. And here I just put an example of the percent methylation from the blood sample from the male and the percent methylation from the blood sample for the female. And remember, they biologically or chronologically, the male was 27 and the female was 47. These are the 6 markers that were already in the literature identified to be correlated with age. And while now, so far, we don't have a statistical model using this chemistry to generate a prediction, just by looking at the percent methylations and the difference between these individuals, especially on these first 2-3 markers, you can see that there are differences in methylation between these 2 individuals. The next step, obviously, would be to to develop this regression model, to do this prediction model, linear regression model, most likely to be able to plug in basically these numbers in a formula, and it would give you the predicted age. Now, moving on to the STR profiles or the STR typing. And that was the most challenging part, because we all know how hard it is to, anyway, sequence repetitive patterns in a sequence. But even though that was challenging, we were able to get up to 65% of alleles were recovered, like this, for example, for this sample. Obviously, there's a lot of room for improvement in this case, but some optimization and probably just increasing the probe concentration for the STR panel would improve the recovery of these STRs, or maybe tweaking some of the probe designs would also help. At this time, we just did a 1-to-1 ratio on these two panels for the CpG and the STRs, so we haven't had the chance to really do more of the optimization, but still from that first run to get that a positive result there, it's very promising, and it would ultimately be much higher than that once we proceed with the optimization. So I kind of was maybe a bit fast, but I wanted to really leave some room, because I know that we're going to get great questions. But before we move to the questions, some of the concluding statements would be that this is really a pioneering effort. It has never been, to the best of my knowledge, never yet been done for a forensic application to simultaneously look at the STRs and CpGs in one run. The key element, besides obviously the library preparation, which was very important, because otherwise we would not be able to look at simultaneously both the genetic and epigenetic markers, was the hybridization capture to just enrich the forensically relevant markers. But further testing will definitely, on much larger number of samples, would enhance its performance and would strengthen its reliability and accuracy. Ultimately, this is a foundation for an innovative solution to save on time, on resources, and on, most importantly, DNA sample consumption, and be able to hopefully solve more cases in the near future. And one of the things that always keeps me super excited is how customizable this would be. So I was talking to my colleagues at Twist and Bimodal, and I said, like, the room to customize this is probably infinite. If we just think of adding potentially more SNP markers that can give you like phenotypic prediction, ancestry, kinship, identity to that panel, because we have room to make it to add more markers, then not only can get human identification, but in case your DNA is so degraded where you don't get a full STR profile, at least you are getting more information that would be gives more investigative leads to different case scenarios. The other aspect of it is now there are studies that look at SNPs that are neighboring CpGs that would be very important for mixtures. So maybe in a clinical context, you're like, oh, why would I have a mixture? That's often not the case. But in a forensic context where you have a stain left in a crime scene, you would oftentimes have a mixture of different contributors. So if you think, for example, the sexual assault case where you have semen with vaginal fluid, that's a mixture. In order to identify the fluid, that's one question, but also to say that the semen came from this individual because you're looking at neighboring SNPs would help in deconvoluting and identifying the different contributors of the different fluid types. So again, the application for this type of solution is really has a lot of potentials. So just adding more of these probes to the panel would ultimately not only look at, again, STRs and CpGs, but potentially add more SNPs for mixtures and for other phenotypic features. With that, obviously it's not a one-man show. It would have not been possible at all without the awesome collaboration we've had. First, obviously, with my biology team at the Center for Forensic Science Research and Education, or CFSRE for short, particularly Lindsay, who had helped a lot in the hands-on aspect and in the idea also of the project and all of that. Biomoro, who really were key also in this project, supporting us and training on their chemistry, Ibrahim with the bioinformatics, Mark on the library, and John, and obviously Twist, without the hybridization capture panel and the design and how professional was working with all of the awesome scientists at Twist, including Rebecca, Paul, and everyone, and AJ, and everyone on the Twist team, and their flexibility, and how efficient and quick they were working with us on designing the panel, and getting also the training done in house. Again, it would have not been possible, so I'm really appreciative of every individual here. Last but not least, Kovaris also had helped in providing us a demo instrument for the ultrasonicator, which we needed it in the prep of the samples. And remember when I told you, remember the OneShot word, and why I was thinking of a name, obviously, for this assay. And that idea came that we're going to call it OneShot, because the first time when I talked about this, when it was still an idea last year, so remember, probably around February of 2023. It was just an idea that I would never thought that's gonna happen. Was in a coffee shop in Philly called OneShot to a friend of mine, and I was just telling her how excited that this potentially can be a great application, hence why the name is OneShot. And I'd love to be connected with you, so if you're interested, feel free to scan the QR code for my LinkedIn. I'm pretty active, I think, on that platform. I'm now ready for any of the questions. Again, thank you all for being here, and thanks for Twist for hosting this webinar.